Yes, good to start.
Yeah, okay. Okay. So, good evening, everyone, and welcome back for the second day of the internship. So, today it's going to be the Metagenomics Day 2 session, and I hope whatever was taught yesterday, it was clear to everyone so that we can begin with today's session. So, can I just get a quick thumbs up from everyone? Okay. Yeah. So before I start, I'll just revisit whatever was taught in the last session, and I'll just talk about metagenomics in few words so that it is clear for all of us, and then we can move forward with today's session. So metagenomics is basically the study of the genetic material that is recovered directly either from any environmental sample like soil, water, ocean, or maybe from a human, an animal, or somewhere like from a particular environment. When you isolate certain microorganisms and study them using a microbial, using a community as a community together, using a metagenomics approach, that is using its genomic DNA, that is what is called as metagenomics. So this was what we covered in the yesterday's session, where we understood everything about metagenomics. We understood the basic workflow, the types of metagenomic sequencing that are done, and why 16S rRNA is very important for metagenomics. So now again, for today's session, I'll again welcome you all to the world of microbes. So again, we'll be talking about this. So before I start, so maybe we can talk about microorganisms. So microorganisms, like I would like to share a few lines regarding microorganisms. So microorganisms are among the oldest forms of creatures that are present on Earth. So they have been existing since like billions of years, even when the humans or any plants or even the dinosaurs appeared. So there's one fascinating fact that like I'm very fascinated about is that these microbial cells in and in on our body, they're approximately equal to the number of humans that are like they're almost approximately equal in number to the human cells. That means that yesterday I showed a diagram as well where the microbial cells and the human cells were compared. So like all of them together contributes like the maximum number, even the presence of them on outside or even inside. So in many ways, we're actually like ecosystem, like our human body, ourselves act like an ecosystem which contains like trillions of microorganisms. So those microorganisms, to understand their classification, their hierarchy, that is why we understand this metagenomics. So that is why this metagenomics was introduced, and that is how we are following it till now, so that we can understand these relations, the phylogenetic analysis, the diversity of these microorganisms that are present. So now we'll start with today's session about the day 2. So day 1 was already completed. That was introduction to metagenomics. So in day 2 session, it is going to be a little more technical as compared to yesterday's or day before yesterday's session. So in the last session, we only understood about the metagenomics, the biological part. So today we are going to be majorly focusing on three topics, that is Linux, Jupyter, and Conda. So we'll go in an order. That is, first we'll understand what is Linux operating system. Why do we use it? What are the advantages? What are the distributions? And what are the basic commands that are present in Linux? After that, we'll move on to Conda. So Conda is going to be a package that will be present in our Linux. And then that is going to be used for our KIME analysis. After that, we are going to understand Jupyter. So Jupyter is basically a platform where you write Python codes. Just not Python, you can even write the Linux command with slight modifications there, and you can execute them so that you can understand and you can analyze. So Jupyter we are going to use so that we can have a list of all the commands that we are using for our KIME analysis. All the commands that we'll be using for our KIME analysis are going to be Linux based commands. But as we need to document it properly, so instead of running it in the terminal, what we are going to do is we are going to run it in the environment, that is in the Conda environment that we'll be creating. And then we are going to run all of that together in the Jupyter notebook so that we have the commands and then we have the output for it. So this is why Jupyter notebook is really famous, because Jupyter notebook has the property where you can run the command and alongside you will be able to see the result as well. So if I wanted to ask, like, how many of you are comfortable with, like, have familiar with Linux, like, have used it before? So you can just raise your hands with Linux. That's it. Okay. So, Vani, can you please tell me how many numbers of hands have been raised, please?
Yes, sure, sure. Three has raised the hand.
Okay. So three people are comfortable with Linux and are like have a basic understanding. Okay. And what about Jupyter and Conda? Have you guys ever used this before, or this was going to be a first time for both Jupyter and Conda? Okay. So three people have raised their hands. That means they're familiar with all Linux, Jupyter, and Conda. Am I right? Can I get a thumbs up if I'm right? Okay. Okay. Okay, Suganya, first time for both. Okay. Okay. So maximum people are going to be learning about it for the first time. Okay. So we'll move forward. So now I'll be taking everything from pure basics so that you can understand exactly what is happening for each and every command. So for Linux, first I think the today's objectives are clear for the study. So now we will move towards the understanding. Tanil, do you have a question? Your hand is raised.
No, ma'am.
Okay, okay, okay. So now we will move further. So now, first of all, how did Linux come in picture? So now we are going to study about a little back-end theory of how Linux was developed. So before discussing about proper Linux, let us learn about the person who created this Linux. It is Linus Torvalds. So this was the guy who created Linux. So before, like, this is a biographical note that I have mentioned here regarding the Linux when it was created in 1991, and it is revolutionizing the open source computing, and then it is also developed like it in a GitHub as well. So now, before we discuss about this, what we'll understand is first we'll go through a story. So I would like to tell you guys a story how this guy created Linux. So Linus Torvalds was born in Helsinki, Finland. Okay. So as a young student, he was very fascinated about computers and programming. Like during the late, say 1980s or something, operating system like Unix was there. Unix operating system was there, but it was very powerful and very expensive. So many students couldn't access it. Among them was Linus Torvalds as well. So Linus wanted a system that anyone can use and improve, but he was not able to afford the Unix operating system that was there. So what he did is in 1991, while studying at the University of Helsinki, he started developing a small operating system as a personal project. So initially, he wanted it to be very simple and better for just his own computer. However, he shared the code publicly on the internet and invited the programmers to contribute worldwide. This simple project eventually became Linux. Now, this was the revolutionary aspect of Linux, and this is and that it was an open source. So it was accessible to all and people could access it. That is how Linux was generated. So today, Linux powers most of the supercomputers, the cloud servers, Android phones, scientific computing systems, bioinformatic platforms. So what is most important to us is the bioinformatics platform. As all of us are from a background of biology, so we are going to use the Linux OS for our bioinformatics systems. So whatever we are going to do is it is going to be based on biology plus informatics. So that is why we're going to use Linux for today's analysis, that is KIME, so that we can understand how the microbes and what and all microbes are present. So we'll run all the commands on Linux for the KIME2 analysis. So now, after we understand the little theoretical concept, so now we'll understand that many people think that Linux is a single operating system. Actually, Linux is more like an engine, what I would like to say. So different communities and different companies build different vehicles around the engine, right? So engine is going to be the core part, and around that you can build whatever you wish to build, and these are known as Linux distributions. So Linux remains same, but the Linux distributions keeps on getting differed. So example of some is Ubuntu, Fedora, Debian, all of this. So Ubuntu is one of the one that like I have used personally for a longer time, where I have run all my commands on Ubuntu Linux. So that is what used to use in my studies and in my projects. So this is just different distributors of Linux. So if I have to give an analogy, like just to understand what I'm talking about is, think about smartphones. So Android is the operating system, but Samsung, OnePlus, MI, they all customize it differently. That is why they're able to sell it differently. So similarly, are the Linux distributions. So they share the same kernel. So here I introduced the term kernel. So we'll understand this term kernel very nicely in the next few slides. So they share the same kernel, but a difference in their appearance. That means the core is same, but the output, like the way it looks, that is a little different. Package management, and target uses. So therefore, this and all is different, but the core remains the same, that is the Linux. So in bioinformatics, Ubuntu is the most popular one because it's a beginner friendly and supports most of the scientific softwares. So that is why I mentioned that I have personally used Ubuntu a lot, so that like it is just one of the versions that is much more comfortable with the bioinformatics analysis. So after understanding the Linux distributions, now we'll understand why are we using Linux for bioinformatics. So specifically, why preferring Linux? So imagine you receive a sequence, like you receive a sequencing data from a microbiome study. Okay. The data set may contain hundreds of millions of DNA reads and occupied hundreds of gigabytes, because there might be millions of microorganisms in one sample. So the data is also going to be very large. So opening such a file in a traditional graphical software becomes inefficient because it requires a lots and lots of space. So Linux allows the researchers to process these huge data sets through command line tools. So that is why we are preferring Linux over any other analysis, because our files are going to be pretty large, and therefore those pretty large files can be analyzed and understood in the Linux using a command line tool. So I will explain all the command line tools that we generally use for Linux. Those are the basic ones that is used for operating. And then later on, we'll move forward when we have to do the KIME analysis. So as I mentioned, it is preferred because it can handle the large genomic data very efficiently. It can support automation where you can write bash scripts, right? So bash is another like language that you can execute in the shell prompt. So here I again introduce a new term, shell. Initially introduced a term kernel. Now I'm introducing a term shell. So we are going to understand this as well. So now we are going to do bash scripting. See, if you have 40 samples and you want to run a same command on all the 40 samples, so if you have to manually do it, you have to run it 40 times. That is going to consume a lot of manpower. But what we can do is we can automate. We can construct a bash script. We can write a for loop in that, and we can directly automate our entire pipeline. That is, we have to just run that script once. All the 40 samples will be analyzed simultaneously. Like not simultaneously, one after the another. When one is done, then it will move to the next sample. So it will keep on doing until the sample ends. So you had to do it for 40 times. Now you only have to do it for once. So now you can see how much time you're saving, right? Before you had to sit on the computer for doing 40 samples. If one sample takes 1 hour, then you have to sit there for 40 hours. But now what you can do is in 1 hour or maybe 10 minutes, you can write a script, you can run it, and all those samples will keep on running, and until and unless they're completed, you can do your other work that is required. So that is what automation here basically is telling. So it is better at resource management, and most NGS tools are Linux based. So right now we are understanding metagenomics. So metagenomics, the tool that we're using is KIME2. KIME2 is also based on Linux. If you're going to do RNA-seq analysis, whole genome analysis, and say transcriptomics, then we say methylomics, all of those tools, whatever commands are used, either they're Linux based directly or they can be run in commands. Sorry, they can be run in R. So that is why we are preferring this, because most of the NGS tools, they're based on Linux system, essential for HPC and cloud computing. So you can run many things at like multiple things at a time, and you can upload and efficiently use the entire Linux system. So now if you have to talk about the features of Linux. So Linux, as I mentioned, it is very widely used and it is very user friendly as well. So therefore, we are going to use Linux for our analysis. So if I have to talk about the features of analysis, so here I've mentioned 6 different features. So we'll visit all of them one by one. So initial is security. So Linux is considered one of the most secure operating system because it uses a strong permission system. Okay, like think of as a research laboratory. So not every person can enter into that room, right? Some rooms may contain sensitive experiments, like we have a like for my laboratory we have a thumbprint. So if I want to open the lab gates, I have to give my thumbprints to so that my the data, whatever is there or whatever experiment is going on, is not leaked out. So every file and folder has permission that determines who can read it, who can execute it, and who can modify it. So I mentioned 3 things: read, edit, and modify. So each file in Linux is going to have a permission. So there's going to be 3 permission: read, write, and modify. If you give all the 3 permission, then you can read, execute, and modify. But if you don't give the 3 permissions, if you only give the permission of read, then you cannot rewrite the file and you cannot execute the file. You need to have all the 3 permissions so that you can work with it. So therefore, the security is very niche feature of Linux. So this basically also prevents any accidental deletion or authorization or unauthorized access that can happen to your data. So it prevents from mishappening as well. So second feature is portable. So portability means that Linux can run on different types of hardware without major modifications. So that means Linux is adaptable. So if it is adaptable, that is why it has been very widely used by people, because it can adjust, right? So similarly, Linux can run on personal computers, it can run on servers, it can run on supercomputers, in mobile devices as well. So because of its portability, therefore it becomes very useful for the bioinformaticians. So a researcher can develop a pipeline on a laptop and then can later run the pipeline on a powerful device or server without changing it much, right? So therefore Linux is a very portable system as well. Next is open source. This probably is Linux most favorite characteristics according to me. Okay, so open source means that the source code is publicly available. So anyone can visit it, anyone can modify it or improve it. So like it is a very open thing that is present. So that is why it is one of the most featured feature of Linux. Okay. So developers worldwide, they continuously keep on updating Linux because it's present in an open source. So because of this, Linux evolves very rapidly because whoever has a knowledge about improving it keeps on editing and putting in the efforts so that Linux can become better and better with time. So most bioinformatic tools are also open source, making Linux a natural choice for these scientific researches that are happening. So now when we move, next is shell. So shell, now I have introduced here once. We'll be introducing again in the next few slides. So just to basically understand, shell acts as an interpreter between the user and the operating system. So I am going to give a command, right? I'm going to write something on the prompt line. So when I'm going to write, then the operating system, now interpretation needs to happen and the result needs to come, right? So whatever I write on the command line, I will show the command line and the terminal now. So whenever I write a command on the commands on the terminal, so what will happen is shell is going to interpret it and it is going to send it to the computer, the operating system, so that the operating system can understand what do I want. So because my language and computer's language is going to be different, so shell is going to act as an interpreter here. Similarly to a translator, if you go to a foreign country, when you can't speak their language, so you need someone to translate in between, right? So that translation job is done by shell in our systems. So similarly, you are going to type a command, shell is going to interpret it, the operating system is going to perform the task and revert back the output. So this command asks Linux to show the output as well. So this shell receives the command and tells the operating system basically what needs to be done. Okay, so it is acting like an interpreter between me and my operating system. Now next is multi-user. So multi-user allows, as the name suggests, multiple users to access the same system simultaneously. So it is acting like a library. Okay, so like in library, multiple people can go and read whatever book they want, right? Nobody's going to hamper or nobody's going to say that no, you can't go, already one person is there inside. So that is why it is also a multiple user, because multiple people can access it. So one server can be shared by many genomic researchers, many microbiologists, many bioinformaticians. All of them can work simultaneously. The generally, when you go into a research setting, where I have been in a research setting, there's a server for a particular lab, and all the people present in that lab are going to access that server for working. So this is a very good capability of Linux that like basically enhances its high performance computing environments. Our next important feature of Linux is multi-programming. So multi-programming is the final feature that we'll be talking about today. Multi-programming means that Linux can run multiple programs at the same time. So Linux can handle multiple tasks simultaneously. That is what it is trying to say. So, for example, a bioinformatician can download the sequencing data, they can run the time analysis, they can edit the scripts, and they can monitor the performance. So, all these four steps that I've mentioned, they can be done at the same time, that is simultaneously. Therefore, it is called as multi-programming. So Linux is going to efficiently allocate the CPU and the memory resources to each task. So this makes it very friendly for a larger scale data analysis. And that analysis can be done much faster because you can target multiple steps at the similar time. So are the features of Linux clear? Any doubts? Am I going too fast? Like is the pace okay for everyone? Can I get a thumbs up? Okay. Like, okay, I see a raised hand. 1 second. No, nobody's raising a hand. Okay. Sorry, I saw somebody raising a hand. Okay. So we'll move forward. So now, once we got the understanding of the Linux system and we also understood the features of the Linux system. So now there are three main components of the Linux system. That is kernel, shell, and terminal. So I have already introduced all these three words. So we'll be understanding what basically these three components mean here. So as we all know that Linux is an open source operating system. So and it contains these three components. So we'll understand all the three of them with regard to an example. Okay. So shell, you already know that it acts as an interpreter. Okay. So now to understand this, so user like me, I'm the customer. Okay. So say we are going to understand with an example of a restaurant. Okay. So I, Anshu, I'm going to be a customer. So that is what I will be equivalent to a user when it comes to the terminal, when it comes to the Linux operating system. So I am the user, that is analogous with the customer. So shell is going to be a waiter, right? Because shell is going to take order from me, from the user, and it is going to interpret it to the chef. That chef, if this and this order is required, then what will act as a kernel? So kernel is going to be the kitchen where that shell is interpreting and going to. So kernel is going to act as the interpreter. Sorry, it is going to act as the core operating system. So with our example, it is going to act as a kitchen. And terminal is going to act as an order, a counter table, or even you can say a menu. So you can't just directly order whenever you go in a restaurant, right? You need to look at their menu. You need to tell from the menu that, okay, this is all I need to the waiter. So similarly, on terminal, you are going to write the commands. So terminal is the platform where you are talking to shell. Okay. Shell is being an interpreter between you and the operating system, right? But how are you talking to the shell? That is through terminal. So on terminal, when you write a command, so wait a second, I'll just open the terminal as well so that I can show you guys. So when you open a terminal, so you're going to write the command onto the terminal. So when you write the command onto the terminal, that command is going to be interpreted by shell. So shell is going to take in that and understand, okay, what the user wants. Now when it interprets it, now it is going to send it to the kernel. So kernel that acts as a core operating system is going to perform that particular task, and when it performs it, it is going to send back the results to the shell, and then shell is going to display it back onto the terminal for us, because we cannot directly contact with shell, like because shell cannot communicate. So shell needs to show and understand. So terminal is going to be the interface where we, the users, are going to connect with shell. And from there, shell is going to take whatever we have communicated to the kernel, so that kernel can understand what needs to be done, and it does that work and gives it back to shell, so that shell can show us the output on the terminal. Is it clear, guys? The people who have an experience with shell, Linux, and all of that, is it clear how the system is working, like terminal, shell, and kernel? Okay. So now this is a basic Linux architecture system. Okay. So in this, we are talking about the same thing, that is the hardware, that is the inner core. Then we have the kernel, then we have the shell, and then we have the applications. So applications is basically the user when we are communicating with the shell prompt. So shell is going to be like in the middle way. So shell is directly communicating to the kernel. Kernel is connected. So if it needs to be stored, it will be stored in the hardware, and the output will be given back. And here at the outskirts, we are there. So users are at the outskirts, and kernel is the core of the particular OS system, and shell acts as a middle interpreter. So this was just a diagrammatic example to show the same thing that I explained in the before session, so before a slide, so that it is clear how the architecture of Linux basically looks like. So now we have understood how the Linux system looks like, the architecture. We have user. User is like giving an input. So we basically have a terminal, then we have a shell, and then we have the kernel. So these are the three main components that give rise to the entire Linux system architecture. So basically, you can see here, is my screen visible? Is my terminal visible? Okay, so this is what we call as terminal, and whatever I'm going to write here, this is going to be called as my command line prompt. So the shell is going to interpret my command line prompt when I'm going to give one. Say I will, sorry. So say I want to write Anshu here, okay? And because Anshu is not a command, right? So shell cannot interpret it. I need to give a command in shell's language. So when I do enter, shell will try to do, but it will say that command not found, because Anshu is not any command that exists. So that is what I'm going to tell. So this is, you write a command prompt on terminal. Now this prompt is going to be understood by shell. If shell understands, then it is going to send to the kernel and give back a response. But here you know that this language was not there. The command is not found. So therefore no result came out. So basically the main thing to understand is here is, this is how the terminal basically looks like. And this is the, after this dollar sign, this is the shell prompt where you're going to write all the commands. So say if I write clear, okay, so I would like to clear whatever is present in the terminal right now. As this is the language that is understood by shell, so what will happen is when I write clear, the entire thing will go off. And this is the result that we see. So it is important for the interpretation of the results as well. And when you send the clear command, shell interpreted it, kernel told what job to be done, and that job was done. So that is how it has been shown here. So this is going to be a terminal screen. So now we understood the Linux system architecture. We understood how the terminal basically looks like. So now there's a very interesting interesting concept. Okay, that everything in Linux is a file. Okay, so like whatever we are going to store, a directory, like a directory, a folder, a file, so everything is counted and treated as files. Say you have regular folders like documents, images, videos, then you have directories that are going to act as folders, hard disk, USB, printers, everything is going to act as a, like you are going to access everything like a file system. So this design make Linux system very simple and powerful, because the same commands can be used to interact with many different system components. Otherwise, you need to use different commands for different system component. So that is why we are saying that all the files and everything in Linux is going to be is a file. So now we'll talk about the Linux hierarchy system. So this is how the Linux file system basically looks like. So there's an entire hierarchy that is present, how the files are stored, and what are the different types of files that are present. So here you can see that there is a root. The slash means a root directory. So everything in Linux, it is going to be originated from this particular root directory. So if I have to give an example, we might all know about phylogenetic trees, the rooted phylogenetic trees. So there's always an ancestor, from that ancestors all the progeny is getting generated. So this is also a little hierarchical file system where you have a root directory, that is everything is going to start from here. Slash means the root directory, all the folds, all the files, folders, software, devices, everything that is present in your system are going to be stored in the root directory. So it serves at the starting point where like somewhere where everything is stored collectively. So every other directory is located somewhere below the root directory. We cannot see anything above, right? Everything is associated with root directory. So root directory is going to be the top level folder, and it is going to be an administrator account. So now from these root directory, you can generate multiple different directories, subdirectories that you are working. So now we have few directories. So these names of these directory almost remains constant because these are the ones that are present very like these are the ones that are always present in the system. So which one to work with and what are the function of each and every directory that is present. So home directory first. So if the username is say student or something, so all the personal documents are being stored in the home directory. So home directory is basically like you create for your work. So after root directory, there's a home directory. In the home directory, you're creating multiple subdirectories where you're putting same metagenomics day one, metagenomics day two. So all that thing has been saved under the home directory. Then we have the user directory. So user directory basically contains all the installed softwares and applications. So many programs that you are going to install on Linux. So where to place these all installed folders or all the installed softwares. So it is going to be stored in the user directory. So you these directories that have been mentioned in this chart, bin, etc, home, opt, tmp, user, and bat. So all these directories are pre-formed. You are not going to form any directories when you download Linux. When you have Linux in your systems, what will happen is all these directories will be already present in your system because they have a specific role. So say user library is going to store all the softwares that you're going to download. So if you're going to download any bioinformatic tools, so it is directly going to go and get stored in the user directory. Then we have the bin directory. So bin directory basically contains any executable commands. So like now we are going to do certain executable commands, like we're going to study some basic commands in Linux. So all these basic commands require the Linux operations. So these commands are stored inside the bin. So whenever you type something, like whatever commands we learn now, so those commands are executed from the location that is your bin location because it contains all the storage commands. After bin, we have an etc directory. So etc directory is going to store all the configuration files. So these files basically are the ones that controls how our Linux is going to behave. So whenever you change a system setting, like you often set update the Linux or upgrade a Linux, so all that information is present in the etc file. So it contains all your system configuration file. So home directory we have already understood. So home directory is the one where we are creating the subdirectories so that we can use it as like for our use. So users directory is going to be in a home directory. So then we have the data directory. This is shown like as a place for like a user bioinformatic data. So in this particular thing, what I'm mentioning here is the data. That is, you're storing the bioinformatics data here. That is a specific directory that you're generating for your data so that your data does not get merged with it. So this data is not generally present in a standard Linux system. These are the ones that are present in a standard Linux system. Okay, so data is another one that you can create like a nested one in home where you are going to store all your bioinformatics data. So then after that um, we have the home directory. Home directory we have already understood where we can use it as a like we can do a nested subdirectory so that we can save our work, save our files, folders, everything in the home directory. Then we have the OPT directory. So this OPT directory is majorly like stores the information about the third party softwares. So many specialized applications are installed here and some bioinformatics tools are also installed. So it basically maintains the third party thing. TMP stands for the temporary variable or the temporary storage location. So if you are installing something and it requires some dependent files, but those dependent files are not required when the system is fully installed. So those files can be stored in the temporary folder. So whatever is needed temporarily is stored in the temp folder. Then we have the user we already understood, which is going to store everything. So, and then we have the var folder, that is the var folder. So var folder is going to contain the variable data like your log files, your databases, your temporary application data. So whatever sort of different log files are there, it is going to be stored in the var data. So this is a basic description of all the files that we have used. So this is going to be the root directory. Bin is going to store all the executable commands. Etc is going to store all the configuration files. Home is your home directory where you can make further subdirectories so that you can save a file, save a folder, save a command file, everything. OPT is going to be for a third party software. TMP is the temporary space that is like present. And then USR is the user related programs. And then we have the var. Var is for the variable data. So most notably for the log files. So is the Linux file system clear to everyone? The hierarchy of the file system that is present? Okay, great. So the thing is, all of them are going to be present under the root directory. Root directory is going to be the main directory. And from there, all of them are going to be generated in this order. Okay. So this is the hierarchy. So now we are going to move on to the commands. So there are certain commands. So I have mentioned too many commands here. So I would like you guys to understand some commands yourself, like as a homework, so that you can also get a hands-on of it. So I'm going to teach you some of the important commands that are present. So first of all, we'll go to a terminal. So this is our terminal. Okay. So now I've just give me a second. Okay, so now I've already created a folder as of Metagenomics internship. So from here in this particular folder, we are going to work. So now the first thing is that first command, the basic command is ls. So ls is going to be the list command. So ls stand for listing. So it is going to list all the files that are present in your particular directory. Just give me a second. Okay, so ls is going to list all the files that are present in a directory. So either it is going to be a folder, a text file, or whatever it is, everything is going to be shown when you write ls. So ls has certain variations, other variations like ls minus l. So ls minus l is going to show you the permissions, a detailed listing. So here you can see that this is what is shown. So I mentioned when I was explaining that for particular file, you need to give three commands, three operations. Like one is read, one is execute, and one is modify. So when it is written as rwx, rwx, and rwx, so r is read, w is write, and x is execute. So when it is given all the three permissions, then it is going to look like this. If any read, write, or execution is missing, so what will happen? This dash is there, right? So it will be present as a dash. So it will be r dash x. That means you can read the file, you can execute the file, but you cannot rewrite it. Similarly, if it is r dash dash, that means you can only read the file and you cannot execute and write it again. If it is rw dash, that means you can read it, you can write it, but you cannot execute it. So ls minus l is going to show a detailed version where it is stored in which folder, what is the size, what is the date and time of creation of that particular folder. So everything is going to be shown here when you do ls minus l. Then we have one more command, ls minus ls. So it is going to show if there are any hidden files. Right now we cannot see any hidden file because there is no hidden file in this folder. If there was any hidden file in the folder, it would have shown it separately. So ls is used to do general listing. So if you want to see a detailed listing, then you do ls minus l. And if you want to show if there are any hidden files, so first we'll do ls minus ls and then we'll do ls minus la. So now here you can see for an hidden file, there are two things. These are whenever you run ls minus la for a hidden file, what will happen is two of these, one dot and two dots, this is basically seen commonly. Okay, so this is one of the basic basic in output that you're going to see when you run the ls minus la command. Okay, so whatever present here, that is that is highlighted, is the files that are hidden. Initially I ran a wrong command, ls minus ls, it's supposed to be ls minus la. Okay, so whatever you can see here, that one dot and two dots, it basically shows that these files are hidden, and these are the permission for it. So you can see here, there is r wx and all of that. So this is basically telling that the permissions are there for the read, write, and execute. Okay, so now what is this dot basically? So this dot basically is telling about your current directory. So if there is any hidden folder of nothing, then it will show a dot that will explain that there is a this is the current directory. Two dots shows that it is a parent directory. So you can move from the front and back to the folder. So it is basically showing you this particular thing. So I hope it is clear what the ls command does. So majorly when we have to list something, we generally just use ls so that it will list all the files that are present in our system. So now after you have done this, now there's another command that is basically shown to do pwd. So pwd is a command that shows that, okay, in which particular directory you are working. So when we use our tools, so it becomes very important for us to understand their path. That is, we need to know where the tools have been installed, because we need to know the correct path of a file or a tool so that we can use that particular tool. So if you don't know the particular path, so wherever you are in the terminal right now, just type in the command pwd. So when you type in the command pwd, it will give you a proper path from your root directory till the current directory. So it is going to tell you the entire path. If you have to come across through anything, or you have to write the path of the current directory, so this is going to be the path. So this pwd command basically tells the path of the directory where you are working in right now. So if you're going to change the directory, the path will also change, because pw stands for present working directory. So it is going to tell a path of your current working directory. So now next thing is that if you need to create a file and a folder. So when you want to create a folder, there is a command in Linux. We generally communicate folder as directory. So say if I need to create a folder, say neighborman. So I will create mkdir neighborman. So mkdir here stands for make directory. So when I do make directory, that means neighborman is going to be a directory. That is going to be a folder. So when I click enter, that means if no error came, that means my folder is generated. So when I do ls, you can see here that a new folder has been created that is called as neighborman. So this highlighted part for my particular terminal shows a folder. The text that is not highlighted, that suggests it's a text file. Okay, so if it is highlighted like this, that means it is a folder. So here neighborman is created as a folder. Okay, so now what will happen is, now I've created a folder. Now I would like to create a file. So for creating a particular file, we generally use a command touch. Same name I'm going to give, neighborman. Okay, it is going to create another system. Now you list it. Now you can see there are two neighborman. Okay, sorry, I'm using the same name. So now we will do is neighborman 1. Okay, so now you can see there is two things, neighborman and neighborman 1. Okay, so here you can see neighborman is highlighted, but neighborman 1 is not, because neighborman is a folder, a directory, whereas neighborman 1 is a text file. Okay, so that is how you create the commands. So if you have to create a directory, use the command mkdir. If you have to create a text file, you use the command touch. Okay, so now next thing is, now I'm going to write something in my touch file. That is my neighborman 1 file. I'm going to write something in that. So now what I'll do is, either you can do is, what you can do is, either you can directly open the neighborman file onto your system, or double tap on it and open it in a notepad and text in. Otherwise you can use some other text editors like nano, vi, and gedit that needs to be installed on your system. So by default in terminal, vi text editor is there. So I'm going to write neighborman 1. Okay, so this is my text file that I created. Okay, so when I do the enter, it is going to open like this. So this is how the vi terminal looks. So you need to understand and you need to learn how to use these editors. This is a separate topic altogether. So right now I'll just write something in it. Just a second. Hmm. So I'll write hello everyone. Okay. Then I'll write how are you, and then I'll just write few letters here. Okay. Okay. And then I will just write something, and then I'll write thank you in the end. Okay. Okay, so this is I wrote, this is my text file that you can see. This is the content that I have written. So what I'll do is now I will save my work and I'll exit. So this is a completely different topic. You need to learn the text editing, like these online tools. Otherwise, you can directly do is what you can directly open it into your terminal, open it into your system. Click on Notepad, and then you can directly edit it. So now we have already written something in my file. Okay, so now I want to display the content of my file. So first I'll clear because it is becoming too crowded here. So do you guys like, can I clear the terminal or do you want to note down the commands? So like, can you just help me out with that? Should I clear? I just got two thumbs up. Can I get like few, like other people also? Okay. So now I need to show the content of my file. So there is a command that you can use to show a content. That means that is cat neighborman one. Okay, so now I'm going to show, when I'm going to write this, the entire file content is going to be shown on your terminal. Okay, you can see whatever we wrote in the file is shown. Now, there are some problems. Sometimes you have a file that contains thousands of pages. So when you do cat command, what will it do? It will keep on going down in the terminal because it is going to display all the 1,000 pages, and it might crash your terminal also sometimes. So there are some commands where you can specifically visualize the initial part of the file and the bottom part of the file. So to visualize the initial part, so to basically write head, when I write head neighborman one, so it is going to show the initial few lines. So you can see instead of displaying the entire file, it showed only few lines in the start. That means approximately 10 lines it showed from the start. And when I'm going to write tail neighbor man again, so that means it is going to show the bottom part of the file. So here you can see, instead of showing the entire file, now I wanted to just see the bottom and the top portion of the particular file. So here you can see that I can see the top portion, and here I can see the bottom portion. So there are three commands for viewing the file. One is cat to view the entire file, head to view the top portion, tail to view the bottom portion. Okay, so now we have understood how to list all the files, how to create a directory, how to create a particular text file. So now I'm going to list all the files that are present. So now what I want is that I want to copy my file neighborman1 into a different file. That means I want to copy exactly the content of neighborman1 into a different file. So I'll use the command cp. It stands for copy. So first file, you have to write the name of the file from which you have to copy, and second is the name of the file into which you have to copy. So cp neighborman1, say I have to copy it into a copy file.txt. Okay, so I'm going to copy the neighborman1 content into the copy file. So now I'll run this command. Now you see I have another file generated, termed as copy file.txt. So now I will view the content of this copy file. So you can see the entire content that I wrote in my neighborman1 file has been copied here. So cp command is used to basically copy the particular file. Okay, so now what I can do is I can copy my this particular file into my neighborman folder as well. Okay, so what am I going to do is that I'm going to give a path to my neighborman folder, because I don't want that file to be copied here. I want that file to be copied in neighborman1. So what I will do is I will say copy cp. What file that I need to copy is copy. I want to copy the neighborman file. Okay, I want to copy the neighborman file. Sorry, that is what is called as moving the file. We are not going to use the command copy, we are going to use the command move, because we are going to move that particular file from one folder to another. So here you can create another copy and you can insert this copied file into the folder. So what I will do is, first of all, I am going to move this copy file, that is copy file, into a neighborman folder. So now I need to write the entire path for neighborman folder. So I'll copy this particular path because this path is still the metagenomics internship. And in metagenomic internship, I have created a folder that is neighborman. Okay. So when you come across the metagenomic internship, then what you'll do is you put slash and then you will put neighborman folder. Okay. So in this particular folder, you're going to move this particular file. So as you can see, no error came. When you will list, you will not be able to see the copy file here because that copy file has been moved to the neighborman folder because I have moved that copy file. So now one use of the move command, that is mv, is to move a particular file. The another command here is, the another use is that you can change the file name by using the mv command. So I don't like the neighborman1 name. So what I'll do is I will do move. I will do neighborman1. Now I want to change this neighborman1 to maybe I'll say neighborman day2.txt. Okay. So now I want to change the name of the file neighborman1 to neighborman day2.txt. So you can change by using the command mv itself. So when you do enter and when you check the listed file now, there is no neighborman1. Instead, it is neighborman2.txt. So now just to confirm that this is the same file, what you will do is you will just see the content. You will just see the content of the file. You can see the same content is present in the file. That means this is the same file which has been renamed to neighborman day2.txt. So CPU is used to copy, MV is used to move the files and to rename the file. So now we move the file to neighborman, right? Neighborman folder. So now I want to see where my file is. So how will I go to the neighborman folder? So that is another command. To change the particular directory, you need to use a command cd. Cd stands for change directory, and you can list as there is only one folder. So what you'll do is you'll put neighborman. So cd neighborman. So now what will happen? From your metagenomic internship, you'll move into the different folder, that is cd neighborman. If you want to come back to the previous folder, you will do cd dot dot. Okay. So you will come back. Now I want to move to my neighborman folder. So I moved back. I will list the files. You can see the copy file.txt is present here. This is the same file that we moved from our previous folder. Okay. So this is how you can change the directory and this is how you can change the file. So now here, if you want, you can move the file back to that folder, or you can rename this particular file, whatever you want to do. So this is what can be done. Okay. So this is like the very basic commands that you can find for the terminal usage. So this is, you are going to use very like vigorously these commands to change the directory, to create a new file, and to visualize a file. So all these terminal commands will be used very prominently. So then there are certain other commands, that is wget. So if you need to download anything from your web server, you will directly use the command wget. So from the browser, it will download onto your terminal. Okay. And then if you have to list some particular, say if you want to zip a file. Okay. So if you want to zip a particular file and unzip a file, so for zipping it, basically you'll use a command zip, and I want to zip my copy file.txt. Okay. So what I'll do, zip copy file.txt. Okay. So now it is going to create. Okay, 1 second. I have to give the name as well. Okay. So what I'll do is, for this particular command that I'm mentioning here is zip. It is basically for a compressing a particular file. So if we have to zip a particular file, so what we are going to do is copy.file. I'll just try this command once. So okay, so for me, the zip command is not installed here, but you can install the zip command on your terminal directly, and you can use the particular command to zip the particular file. So if I install now, it's going to take a little longer time, but if you install it on your system, it is telling you how to install. Okay, sudo apt install. So when you use this command to install the zip file, so you can install it directly and then you can perform the zipping thing. Okay, so now what you can do next is, now whenever you start using your terminal, so these were the basic commands. So now when you start using your terminal, so what you are going to do is that first of all, the first thing is going to be sudo apt update. So you have to run this command sudo apt update, and when you click enter, it is going to ask for your password for your Linux. So you have to give the password, and now you have to update your system. So it might take some time, so we'll ignore this. In the meantime, just listen. So you will update your system before you work, so that the system is up to date and you can directly use it properly. It is showing that 24 packages can be upgraded, right? So now next command that I'm going to use is sudo apt upgrade. I'll just write here and show sudo apt upgrade. So when I run this particular command, now my files will start getting upgraded, that all the packages that were present now will start getting installed that are dependent. So before you work in your terminal, these things should be done completely. So is this clear, guys? Is this all of this clear? How, what are the basic commands that we can use on Linux, and what are the like update and upgrade pattern of Linux? So Linux everybody has, so this is the basic thing. So all the commands, command line prompt, the guys who are beginners and who haven't like used Linux at all, and this is the first time seeing, is it clear to you guys? Do you have any doubts? Okay. So now there are certain commands as well that I would like you guys to perform it on your own, so that it becomes easy. So for you to understand as well, and once it is understandable, then we can get a hands-on of it. So it is very important to learn these commands. So the best way to learn is that you should just keep on running it again and again. So when you use it very commonly, you will become habitual of it. So you don't need to learn it like by hearted. So understanding it will be better, and performing it regularly will make it leave an impression in your brain, so that you can understand and you can use it in a conscious way whenever you're doing it next time. So this is all about the files and navigating and all of that. Oh, one command I missed, that is deleting. Okay, so when you have to, so when you list here, so now I don't want this file. Okay, copy file.txt. So what I'm going to do is I'm going to delete it. So delete, there is an rm thing. Rm means remove. So rm command is going to remove my file. So I'll do rm. Now I'll list it again. There is no file present because I removed my file. We should be very careful by using rm command, because everything in Linux is a file, and if you try to do something like you use Linux rm like very like not properly and just randomly, there's a chances that your entire system can go, because you will deleted the entire bash rc file, the entire system's content. So your system might crash, so your terminal might not stand because you have deleted everything. So like the people who have used Linux, have you ever faced this problem where you have deleted the file itself? Any one of you? Oh, nice. Everybody is a very good user. I myself have like when I worked in the Linux for the first time, I removed my bash RC file and my entire terminal went. So then I have to fix it all back again, and that's why I learned via hard way. But still, like everything is a file here, so you need to very judicially use RM. So that rm minus rf command, other command is also there, that is rm minus rf, that is you're removing it by force. So these commands should be used very judicially, because these can lead to the removal of the entire content of your system, and all the files that are present in your Linux root directory, everything will be gone. Like you have to reinstall the everything, so it is going to be a very big task. So just use this particular commands judicially. Whatever you want to delete, that time only you use these commands. So these are the basic commands that are very rigorously and very like routinely used in Linux to move from one directory to another and all of that. So now we have almost cleared with the Linux. So now we have understood everything about Linux, that Linux directory, Linux commands, what and all are used, and the Linux hierarchy system, and Linux architecture. All of that we have understood properly in detail. So I just want to ask once, everybody who is who has joined this work internship, do you all have a Linux system with you in handy? Everyone?
I do have Linux system.
Okay.
I have installed it in my Windows only. Like,
Haha.
yeah, but I can handle because my RAM is around 32 GB in my computer, so I
Yeah.
can manage. Yeah.
Yes, yes, that's pretty good. So like you have installed it as a subsystem, right?
Yeah, as a subsystem. Yeah, yeah,
Okay, okay,
yeah.
great. So other people, do you also have Linux handy with you?
Yeah, I have also one, but it's in the laboratory server.
Okay, okay, yeah, absolutely fine. It just should be a like server also is fine. So other people, okay, okay. So can you people please raise your hand who and all don't have? Oh, I think there might many people who don't have Linux on their systems. Okay. So first of all, yeah, okay. So okay, Bhavani, can you please tell how many people are there who have raised their hands?
Sure, I'm sure.
Yeah.
I think 3 plus 1, 4.
Okay, okay, four people. So, four people don't have Linux systems. So, okay, you guys have your Windows laptop, right, with good RAM?
Yes, ma'am.
It's 2.
Okay,
How about
okay,
I have 1 to lower the hand? Now it's 2 in number.
okay.
Now,
So, now what I will suggest you guys is you can use the online servers and the online protocols that are present for downloading Linux. So, what you can do is you can download WSL, that is Linux subsystem on your Windows. So, you will have the PowerShell in your laptops, the Windows users. So, what you can do is you can go to the online platforms and you can search for the downloading commands for Linux. It is not going to take much time, maximum 1 hour or 1.5 hours depending on your system. So what you can do is you can ask how to download WSL. So once you download WSL, you can get the terminal like this. So it is, I have also downloaded it as a subsystem. So that is why I wrote WSL in the start when I opened the PowerShell. So what can you can do is you can also download it similarly. So next session, before the next session, you guys can try downloading, and then what you can do is if you guys have any doubt, you can post it on the groups. I might help you with guiding if you face any problems. But the installing entire protocol is present online, so you can directly go and access that and install WSL systems. So majorly you search for WSL, okay, because it is going to be a subsystem that you can download easily on your systems. Once you have downloaded Linux, then we are going to download Conda. So how many of you have Conda installed? Like, is it clear how to download Linux? Like, there are some commands that you need to run. So that will be as that we have not covered in our session. So you can directly access it through online. And if you guys are not able to, then maybe you can connect back so that I can try giving you some material that I have. Is it clear?
Ma'am,
Okay.
what is the minimum configuration requirement for installation of this Linux?
What is the minimum?
Configuration of the system, like RAM and
Like, it's not that much. A basic system can also handle Linux. It should have a storage, like your system should have some storage, because the WSL system might take some storage of your C drive. So if you have enough storage and basic RAM, like anything more than 4 GB is good for the Linux and all.
okay.
Okay.
Mm.
Yeah. And how many of you have Conda installed on your systems? Okay, only one person. So other people, I'm assuming you haven't installed Conda. Okay, Divya as well. So other people who had Linux, but Noreen, do you have Conda
I
installed like you
see.
mentioned?
I've been using Linux. I have Conda in my system, but I'm not too sure whether I've installed it or not. I have a
Okay,
Conda exe file, huh?
okay,
And all the support files are there. I'm just checking on my system. Support
okay,
files
okay.
are there. Yeah,
Yeah,
but
yeah.
is it a mandate while installing the Linux? Is I really don't know. Huh?
Oh, okay, okay. Yeah, like
Uh,
it doesn't come with the Linux environment. You have to
ah,
download it
yeah,
separately. Yeah.
yeah. I must have downloaded, but I'm not installed and used.
Yes, yes. Okay, understandable. Okay,
Yeah,
understood.
yeah.
So people who haven't installed Conda, I am going to give specific commands for installation of Conda. Linux was a basic requirement, so I was assuming that everybody has a Linux environment and a Linux subsystem. So you guys can start installing Linux, and after the Linux installation, what we can do is we can install Conda. So we'll study about Conda now, and then we'll talk back again about the systems that needs to be installed properly. So for Conda, I have given proper commands that you need to install. And like the way I installed in my system, I have given the same exact commands. But if you guys face different sort of issues, like different problems, then maybe you need to look up as well that what problems your system is facing. But whatever I have given is from a basic installation steps that you can directly follow, and you can download Conda in your particular Linux systems. So before we move on to that steps, first we'll understand what is Conda. So we use multiple tools like Kime2, FastQC, like SAM tools, BAM tools, all of these tools are needed. And then we need a way to install them as well. So from our terminal, we can directly install these tools as well, like by sudo apt command that terminal gives. But the Conda is a better way to install all of this. Okay. So Conda here I'm going to tell it is an open source package and a dependency and an environment management system. So in simple terms, Conda basically helps us to install softwares, install required libraries, manage any software, create different working environments. This is the best feature that is about Conda, and therefore it basically avoids if there is any conflict between different packages. So it is one of the most widely tool used for bioinformatics because you can download multiple dependencies in different environments. So if I have to tell it very simple in a simple word, so when you have different tools, say Kime2. So Kime2 requires a specific version of Python that is not that is generally we don't use for a normal working, because that is what Kime2 requires. So when we try to install Kime2 directly onto our systems, what happens is there's a clash. Kime2 will not function because of the outdated Python version. It requires a specific Python version to Okay. So like there's no link to Linux installation. There are few commands that you need to run in your PowerShell as an administration so that you can install Linux. So I would suggest you guys to try once. And if it doesn't happen, maybe I'll try to share a small like cheat sheet kind of a thing where I'll write few commands to install Linux. Because there might be some errors that might come depending on your system. So you need to like look into that. You can try putting into the AI servers and you can check that, okay, at this particular point, this error came. So once try this, if it doesn't happen, then I'll give you the particular commands that is required, but that might not work properly with your system as well. So you need to be like a little flexible about that. But just try once and let me know if it happens, it's good. Otherwise, I'll try to give you commands. Okay, so where were we? Okay, the Linux. Sorry, the package. So Conda. So what happens is the Python that is required is different. Okay. So therefore, what happens is that there's a clash between them. So now what we do is we create different environments in Conda. Say I want to do Kime analysis, so I will create a Kime environment in Conda. So that Kime environment will contain all the dependencies that is required for Kime function. So other environments, when you exit out that environment, that a particular dependencies are not present in a global format, but they're present locally in that particular environment that you created. So why Conda is beneficial? That is, you can create multiple different environments, and in each environment you can install a particular tool. For that particular tool, you can install the dependencies in that particular environment, and you can directly work there. So that is a very important feature and a very interesting fact about Conda. So that is how basically it is an environment management system. That is why it is named so. So next, it is basically a package manager. So because a package is simply a one software program in a library. So but it helps to manage all the packages that are installed in different environments. Therefore, it is also termed as a package manager. So a package, like there are some things like Python, NumPy, Pandas, all these packages are present. Instead of downloading and installing each package manually, Conda can install them automatically in a single command. Therefore, we can also term it as package manager. So these are the dependencies that are present. Okay, so that is what is required, and that is what makes Conda very beneficial. So one thing I would like to mention here again, that the kind to analysis that we are doing, it requires a lot of dependencies. Okay, so those dependencies should be only installed in that one environment so that it prevents any clashes from the other environment. Okay, so now there are certain features that I've mentioned here. First is cross platform, that is, it can work on Windows, it can work on Mac OS, it can work on Linux. So it is not just specific to Linux, but it is also like present for other capable running platforms as well. Then it can support multiple languages, that is Python, R, Ruby, all these different languages. It can support all these languages. So basically, Conda is available in two distributions. One is Anaconda and one is Mini Conda. So Anaconda is the full version, that is like with all the pre-installed packages. Mini Conda is basically a lighter version if you have a very less storage space or something like that. So you can install Mini Conda. This is a lightweight version with only Conda and essential tools. So this is the two types of distributions that are available for Conda. So environment management that I've already talked about. So it is allows basically to create multiple different environments and manage them simultaneously. You can create multiple different projects and then work on them simultaneously, because there's the conflicts that can happen between the two different environments will be prevented, because all the dependencies will only be installed in one particular environment. Therefore it is not going to hamper the functioning of the other environments or the other packages that have been installed in the different environments. So similarly, so Conda, this is just a repetitive slide that I've mentioned by focusing more on the key functions and common distributions. So I've already talked about the common distributions about the Mini Conda and Anaconda. So if you have a bigger, like you have a more RAM and a good system, then Anaconda suits well because it is going to install all the packages and everything. But it is common, like anything you can use. Mini Conda and Anaconda both are going to be perfectly fine. So in bioinformatics, we're using Mini Conda because it is lightweight and whatever we need, we can keep on installing. Anaconda becomes very big, so that is why we are using this itself, so that we compromise there. So some key functions that I would like to mention about Conda is that it can install complex software packages. So all the bioinformatic tools and libraries like KIME2, BioPython, Pandas, NumPy, all of these can be installed directly and very easily in Conda. It can manage library dependencies. That is, many programs require very specific versions of libraries that I just mentioned, like Archive. So if Conda automatically installs the required dependencies, then that's why it becomes compatible. And whatever dependencies is required, it installs and it prevents that clashing from the other environments. So that is why it is preferred over other environments and other for basically for the bioinformatic tools. It can create isolated environment. Again, the same feature of these isolated environments are the features or the packages that are installed in one environment will remain in that environment and is not going to clash with the other packages. Then you can run the bioinformatic tools seamlessly. The tools can be installed and executed very easily, and they can be only present in a dedicated environment. So if you have an environment, you activate that environment, you go into that environment and you run the commands for that particular tool. So say if I've installed FastQC, so when I go into the into the environment where I have created for FastQC, in that I have installed the tool FastQC. So whenever I go into that environment, then only I can use the tool FastQC. Whereas if I'm present in another environment, that means I cannot use that tool because that tool was only installed in that particular environment. Because of this specificity, Conda is very useful. And the main reason here is that some tools that requires too much dependencies, it is better to install them in Conda rather than scattered in your terminal, because Conda gives a very targeted environment for that particular tool to function. So is it clear, the information, the understanding about Conda? Why are we using Conda and why is it beneficial? Can I get a thumbs up, please? Anybody has any questions till now? Okay. Okay. So these are the commands that you need to use to install Miniconda. So see, I mentioned, right, wget. So you have to copy paste these commands directly onto your terminal. And when you copy paste it onto your terminal, you can directly download it using these commands. So wget, this is the repository or the Linux bash file for the installation. So you can download this installation file using the wget command directly in the terminal. It is going to access directly from the web browser and install it. Then you will run this downloaded file, that is going to execute it, and then you will activate it from your bash rc. So now bash rc is your like bash rc is a very niche file that is present in Linux. So bash rc basically contains paths to all the files and the tools that are present, and if bash rc is deleted, your system goes. Okay, so this file is important to keep all the paths and all the files that are present so that the Linux keeps on working. And then you can check using Conda version. So when I did this particular thing, I faced a problem where I wasn't able to directly, where my bash RC couldn't show the path for my Conda. So if that happens for you guys as well, so what you can do is you can run this command after you run the installer. So after you run the installer, you can run this miniconda bin conda command, and after you run this, you again source your bash rc. Then that error will be gone. Initially what happened is, in this particular thing, they will ask you a permission that should they add the path of the conda to bash rc, but it has been automatically set to no. So because of that no, what happens is the path is not added to bash rc. So is anybody feeling like, what is she telling? Path, bash RC, like, is it too much of jargons that I'm throwing? Is anybody like, are you guys clear with it, or should I like go a little slowly and explain in much more detail? Okay, clear. That's great. And everybody else, I just got one response. Yeah. Okay. So this is what it is. Now, after you install the Conda, now you're going to basically update the Conda. That is, you're going to update this Conda. If update happens, well and good. If update doesn't happen, so this is a troubleshooting commands. You can run these, and then you can do the Conda environment check. So, and then we're going to install Mamba, because Mamba is by using Mamba, we can directly install Kind2, and that becomes pretty easy to install as well. So now I will make you guys understand. Sorry. Okay, so now you can see here base written. That means I have already installed Conda in my system. So base means that you are present in base environment. So now if you have to see how many environments are there, do you do Conda environment list or Conda list environment? Okay, so when you do this, it is going to list all the environments that are present. I always confuse between this command, either it is first or next. So I'm sorry about that. So this is this is basically the base environment, and then we have the KIME environment. I have already installed KIME, so you can see that my KIME environment is present. So when you use the command conda environment list, it is going to list all the environments that is present in your system. So I had these two environments, so it is showing me these two environments. So now when I have to activate one particular environment, I use the command conda activate and the name of the particular environment. See, for the for the time being, I'm going to activate my KIME environment. So when I'm going to activate my KIME environment, this is going to be like how it is. So now, before, you can see here, I was seeing base, right? Base means I'm in my base, the common environment. Now, when I'm going to activate my KIME environment, and from the base, it is going to move to the KIME environment. Now you can see here KIME is written. That is, I'm no more in my base environment. I'm in my KIME environment. So you guys don't have to worry about the installation of KIME right now. Today's concept was about Conda, Linux, and Jupyter Notebook. So I have only mentioned about installing the Conda and then the Jupyter Notebook. I was hoping Linux everybody has. And then KIME is going to be covered in next session, where I'm going to tell about the installation steps, everything step by step. Will move forward for the installation of KIME as well. So right now, just understand how Conda works, okay? So now I've activated Conda. Now I want to deactivate this. So what I'll do is Conda deactivate. So when I do deactivate from my So when I'm going to use this command conda deactivate, so what will happen is it is going to, I don't know why that error happened. So maybe I put a wrong spell.
Spelling was wrong. Yeah.
Okay. So now when I wrote conda deactivate, so it is going to deactivate the KIME environment and I'll move back to my base environment. Okay. So that is how you basically move within the environments in conda. So I'm going to again activate conda, activate my KIME environment because I want to stay in that. So here, so all the dependencies that is required to run the KIME commands are going to be installed here itself. Okay. So you don't need to install anything else in this environment. So in my KIME environment, all the things will be installed properly. So now when you move on to the KIME environment, there are certain plugins that are required. So there are certain commands like plugin. I would like to just say that that is a function. For the time being, we'll just understand plugin as a function. Okay. So that function is present. Everything is present here. So now we will understand what and all plugins and commands do we have. So we'll use a tool KIME help. It is going to show everything what the KIME command line commands contain. Okay. So version is going to show the version and help is going to show this message. So this message shows that what and all commands are present. So these are all the plugins that are present in particular KIME analysis. Okay. So we'll understand plugins in more detail in the KIME session. Right now, I'll just make you guys understand that these are the plugins. So whatever function you need to perform in KIME analysis, for that, all the plugins have already been installed in the KIME. So in the package that we have installed for KIME, all these have been already installed. So we don't have to worry about it. And therefore, this can already be seen. So whatever we require is already present. So we'll just look into it. We'll run it into the command and we'll get the outputs. So this is how basically you are going to use conda. So therefore, my KIME environment is only associated with my KIME analysis. So KIME will only work when I'm in my KIME environment. So I'm going to just show you one more thing. When I deactivate my environment and I move to the base environment, so now when I'm writing KIME help, okay, I'm right now not in my KIME environment. It is going to show that KIME is not installed. So this is the best concept about conda, as the clashing will never happen. In the base environment, there will be nothing, no tools of KIME. All the KIME tools will only be present when I move on to my KIME environment. So that is how you create isolated environments in conda, and that is why we are using conda, so that we can segregate between our different uh, environments, and only those tools can be installed in a particular environment with only those dependencies that are required to run that tool. Therefore, no clash happens. So is it clear what is conda, how to install conda, and how to basically activate and deactivate environments in conda? Only 4 thumbs up, guys. Okay. Okay, great. So now,
Ma'am, I have a question.
yes, Tanvi.
So, if I'm inside an environment and
Yeah.
some tools which is common, I have already installed in the base environment. So, is it
Okay.
necessary to install in that particular environment?
Yes, it is necessary to install because every environment is isolated.
Okay. So, it cannot be linked between the base environment and the other environment.
No, like maybe there might be a linking concept, but for the
Yeah.
time being, it should be separate. Like you can do a nesting. That is, you have an environment, you can create a nested environment in that. So that and all is there, the linking part, but it is not going to be connected directly because these are isolated environments.
Okay.
Okay. So now there are some commands, some basic commands for conda. So conda information you can get by using conda. So once everybody has installed these commands, I would like you to run all these commands so that you can understand what is the basis of conda. So this is the basic commands, okay? So if you have to update your conda version, you use conda update. Then if you have to update anaconda, so use conda anaconda update. So all these are for different. So if you don't have anaconda, you don't use the anaconda commands, only use the conda normal conda commands. So to install any package, you can directly use conda install and the name of the package. But as our KIME has a little much more dependencies requirement, so we require a little longer command for that to install the packages. So then you can understand the version where you can see conda search package name with information. So if you want to create and install something, and this is the help section. So whenever you do dash dash help, it is going to give you all the help and whatever command that needs to be run, what is the syntax. So it is going to give you syntactical help regarding that and what are the inbuilt tools for that thing. So all that information can be you can get when you are running the help command. So then when you work with the environments, then these are the certain commands that you can understand. So I just showed about activation and deactivation. So all these commands you might not use that much because you only use the particular activate and deactivate for moving on to the different package. So, but as a homework, everybody can try running these commands, not running, just understand once what is happening here. So it is just regarding the environment that you have created. So each command will be a little different and it's going to give a little different type output. So this is basically telling how to create an environment, how to delete an environment, how to activate an environment. All of these things are mentioned in these commands. So is Conda clear to everyone?
Yes, ma'am.
Okay, great. So now we will understand Jupyter Notebook. So just give me 1 second. Okay. So now we'll understand what is Jupyter Notebook. So Jupyter Notebook is basically a platform where you can run the Python scripts and that too you will get an interactive output. So generally what you do is you run the entire code and then you get the output. But in Jupyter Notebook is you run one code and after running that code you will get an output for that so that you can visualize it simultaneously. So it is an interactive and a visualization format. Okay. So in Jupyter Notebook, like as we suggest, Jupyter Notebook is an open source interactive platform where you can code, data analyze, and visualization. So it will allow you to create and write a code, execute it simultaneously, and then view the results in a single document. So otherwise, if you have to run it in a terminal, you have to create a Python script, you have to execute the Python script, and then the results will be same. But in Jupyter Notebook, what you do is you write the code, you execute it simultaneously, and you see the output as well there itself. So therefore, Jupyter Notebook is very commonly used, and the most commonly used language with Jupyter Notebook is Python. Although it supports many different languages, but Python is the major base of the particular Jupyter Notebook. So one of the biggest advantage here is that you can write the code, you can get the output, and even you can write text, document the stuff as well in the Jupyter Notebook itself. Like you can create a full log file in Jupyter Notebook where you have a heading, then you have the explanation of the code, then you have the code, then you have the output of that code. So everything can be generated in one Jupyter Notebook. There are some key features of Jupyter Notebook, that is interactive code execution. So codes can be run and one cell at a time. So one cell you run one code and that code is going to be giving you the output. So therefore debugging becomes easy because each code you run, you understand the output, and if it is wrong, you can correct it individually. Then we have data visualization. So data visualization it means that it supports the plotting of graphs and chart and everything. So when you plot, when you do a Jupyter Notebook, you will give a plot command where you can plot a particular curve. And when you see after you plot at the bottom of that line only, you can see the plot. So it is easy to understand and visualize as well. And if there is any error as well, you can just make edits in that particular line of code, and you can see the changes in the image that is generated. So along with that, it is also integrated with NumPy, Pandas, and BioPython. So all these libraries have been compatible with Jupyter Notebook so that you can do data analysis part in using Python. That is, you can do data editing, data understanding, and data cleaning. All these things can be also performed using Jupyter Notebook. Then we have step-by-step workflow documentation. Because when you're running everything in Jupyter, everything is getting documented command by command. Whatever command you have written, you are writing a comment, you're writing a result. So everything will be written and it is going to be well documented file. It is very easy and easy to share and reproducible. That is, you can directly share the Jupyter Notebook file to the other person and they can directly visualize it in their Jupyter Notebook exactly in a similar way, the way you saw it in your system. The command, the images, and all of that. So everything can be shown properly in a flow. So that is why we use Jupyter Notebook, and it stands to have like good features, and it is very interactive for visualization. Then the bioinformatics application for Jupyter Notebook is like the data processing, the statistical analysis, running bioinformatic pipelines, the same thing. Whatever you write here, you do this for bioinformatics, the same things will be considered as for bioinformatics applications. So you can write the code for data processing, you will get output, you can perform statistical analysis, you can run the pipelines. We are going to run the KIM2 pipeline in Jupyter Notebook itself. So when we run it, for after each step, whatever outputs or graphs we get, so we can visualize there, the visualize that as well in there only. And then it will create a documented file so that we can keep it further, so that we have the entire commands documented in one single file. So the standard workflow for a Jupyter Notebook is you write a code, you execute it, you analyze the data or analyze the report that you get, you visualize it, and then you document the thing. So everything happens in one single file. So now in Jupyter Notebook, there are basically two type of cells that you will see once I'll open it. So one is going to be a code cell where you write the code, and one is going to be a markdown cell. So markdown cell is basically you write it in markdown syntax. When you write it in a markdown cell, that will act as your text. So you can write in that which font color you want, which font size you want, all of that you can mention. And once you do that, you will get a proper very like proper text output, so that you can understand uh, that this is the heading, this is the explanation of it. So it will come like you have made a report. Say you have made a report with the title, heading, and everything. All that you do in your text files, you can do in your markdown cells here in the Jupyter Notebook. Okay, so is it clear about the structure of Jupyter Notebook, about what Jupyter Notebook is and how and what and all it contains? Anybody has any doubts? Okay. So now there are certain commands. Okay. So these commands we just understood right now, right? All these commands. So we are going to run these commands in Jupyter Notebook now. As these are all Linux commands, but we're still going to run it in Jupyter because we want to understand the Jupyter Notebook as well once. So all the commands for the Linux also, all the commands for KIM2 also are Linux based. That is, they're all direct terminal commands, but we are going to run it in Jupyter Notebook because we want to see how Jupyter Notebook works as well. So now when you install KIM, so I first have to activate the environment. Okay, Conda activate KIM. So when we install this KIM environment, our Jupyter Notebook itself will get installed along with the packages that are present. And once you activate your KIM environment, you can also access your Jupyter Notebook. Okay. So now Conda, you can see my environment is active again. So now how to access the Jupyter Notebook? You just write Jupyter Notebook in the terminal. So now this is very simple. It will show different local host web server where you can directly click and open it into your web browser. So this may be any of the URL you can copy from here. And once you copy, you have to go to your browser. When you go to your browser, what you will do is you will paste it here on your search bar, and you can see that the Jupyter Notebook is created. Okay. So this is how basically it looks. So whatever directory we were in here in my terminal, that is inside my Neighborman internship, here in Neighborman directory, my Jupyter Notebook is created. So it is going to follow the same path. So whatever files you would have put in Neighborman, your Jupyter Notebook will be able to access all those files. And whatever you're going to save in your Jupyter Notebook, here in the web browser, it is going to go and get saved in the Neighborman folder. So is it clear, the saving part? Any doubts in that? Okay, just for a quick question, I want to ask, if I would have opened my Jupyter Notebook from Metagenomics internship, so in which folder my data will get stored, whatever I have executed online?
It will be
Maybe.
inside the Metagenomics internship.
Yeah, that's correct. Okay. So the people who are new to all this, are you guys clear with it? I don't remember the names, but there were few people. I guess Praveen, you were for like you haven't used Conda and Jupyter Notebook before, right? Okay. Suganya, are you clear with this?
Yeah, I'm following it, but with
Yes.
hands-on, I can get better understanding
Yeah,
as
yeah.
well.
Absolutely. So, like for the time being, just like I would like to say that just focus on this, and once it is done, then you can do hands-on. And if you have any doubt, you can directly contact. Okay, so that's not at all a problem. So once the this thing is clear, so I just want you guys to have a basic understanding in the session so that you can go and practice it back. So I hope that basic understanding is getting developed, right, for everyone.
Yeah.
Okay, great. So now everything that is going to be made here is going to be stored. So now what we'll do is we are going to create a new Python 3 file. So it will open in a new tab. Here, this is how it basically looks. So here you can write the name. So say I want to write the name as basic Linux commands. Okay. So basic Linux command. This is the name of the file that I have stored. So now my name of this file is basic Linux commands. So now what it will show is whatever is present in my current, like I can run all those commands that I was running here in the terminal now in my Python notebook. There I couldn't make a document of it, but here I can make a document of it. So now, but the only catch here is in that you wrote it directly like pwd, but here as this is working in a Python language, you cannot write the Linux command directly like this. Rather you need an exclamation mark in the start so that you can this Python code can understand that it is a Linux command. So when you run like this, so how do you run a command here? So you have to either you click on run, okay, or either you do shift plus enter. So when you do shift plus enter, the command will run, and this is one cell of the code, and this is your output command. Okay. This is your output command. This is your output result. So we search for the current working directory. Now it showed. As I mentioned that the current working directory is going to be the same as in the Linux from where we opened our Jupyter notebook. Okay. So this is our current working directory. So this code of cell is the code cell. So there are two types of cells. One is a code cell and one is a markdown cell. Okay. So one now, right now, which we are working in is the code cell. So we'll run another command. Let's say this ls. Okay. So you can see that this is the file that is stored in our particular folder right now, because this is the file we just saved, right? Basic Linux command.py. So this file has been saved right now in my system, and that is what it is showing here when I do the command lists. So from here also you can create a folder in your Linux. What it will happen is like you'll do mkdir, and name of the folder will be result. Okay. So now it will create a folder. So now anybody, now I want to list the files again. So what command am I going to use now? I want to see that my folder has been created or not. So I want to list the file. So what command should I use?
ls.
Okay, great. So ls. So now you can see that another folder has been created, that is result. Okay. So now I want to go into this result folder. So what command should I use to go into the result folder? Maybe anybody else can answer for this tunnel. You have been very nicely answering all the questions. Maybe we should give some chance to other people as well. Okay, so anybody else, I would request somebody. Tunnel is giving all the correct answers, guys. You should also try once. So I want to change my directory now. So how will I do that? Correct. Correct, Nilanshna. So we're going to do CD, and then we can move to the result directory. Okay. So now I want to check in which directory I am. How to check? Which command, guys? Suganya, maybe you can try this. Now I have moved on to my result directory. Now how will I check that? Am I correctly in my result directory or not? So which command is used to see the current working directory? Anybody would like to answer this question? The command is itself written on the screen. Correct, Nilanshna? Correct, Divya? Okay, we'll write pwd. So now you can see here. So we haven't moved into our directory result. Okay. We are still in the same directory. You can see here, right? So that means that you cannot move into the directory. Rather, you can just create here and that's it. So like you're not able to move into the next directory. That is what you can observe when you write the current working directory. So now what you can do is, now we can create a file as well. So you'll do file, this thing, exclamation mark touch 1.txt. So once this is done, so now when you do ls, you can see that 1.txt file is also created here. So this is basically showing that all the environments that you can see here, all the commands, these are all Linux based commands, but you can run it here in the Jupyter notebook because you are able to, we are putting exclamation mark. So all the Linux commands can be run in this notebook because of an exclamation mark. Okay. So now before that we also checked, right? Kime help. Just give me 1 second, guys. Okay. So the same command, that is Kime help, that I ran in the terminal. So what I'll do is I'll try running it here as well. So we'll do Kime and help. So this is like here you can see that it is showing the entire Kime options what we just saw, right? So everything can be seen here. So this is how the output basically looks. What we were basically seeing in the terminal before, now you can see it here. So now when we save this file, you can document this entire thing. So we can run the same commands that we run in the terminal here. It's as well in the Jupyter notebook using an exclamation mark, and directly you can see. Before, when I ran all the commands, I cleared everything, nothing could be seen, right? But here I have documented each and every command. I can even write a comment here. I can just put a hash. I can write to check current working directory. Okay. So I've added a comment. That is, for this command, I have given a small explanation that if I have to check a current working directory, I can use the command pwd. Okay. So now we'll just understand once again what is a markdown cell. Okay. This is a markdown cell. So I wrote hello here. Okay. So when I did an enter, you can see it will come out like a text. You saw? That is what is a markdown cell. So when you write hello here in the code cell, it will show an error because hello is not a command. Hello is nothing in Python. But when I convert this to a markdown cell, I can write whatever I want as an explanation. So I'll write, hi everyone, I am unsure. Okay, when I wrote this and I'm doing enter, so you can see everything is written as a markdown cell. So this is going to be like if you want to create a file, or if you want to create a log file, and if you want to write some information about your command or about your results. So you need to give an explanation. Like you need to give a like description of your image. So after your image is generated, you can create a markdown cell. So in the markdown cell, you can write a description. So this is how these two cells are used, code cells and the markdown cells. So is it clear to everyone what and all cells are present in the Jupyter notebook? Okay, great. So these are some of the basic commands that I ran here, and that's it. What I can do is, now this is automatically got saved. Okay, what you can do is save and checkpoint. Okay, now you have saved it. Everything, whatever has been written in this file, has been saved in your folder, that is Neighborman. Okay, so everything is saved here. So whenever you open this file here, you can see that the entire similar file can be created. A similar file will be generated. Okay. So now when we go to the terminal, when we want to exit this particular thing, what we'll do is we'll do Control C. Okay. So now after you do Control C, it is going to ask, shut down this notebook server. So what we'll do is we'll give yes. So yes means it is going to, okay. So here we took some time, so it continued the operation. So we'll again do Control C and we'll press yes. So now it has done. So something went wrong and somewhere it showed so many errors. Maybe I pressed some wrong key. So it is confirmed it happened. That is okay. So we'll do clear. And now we'll list the files here. So you can see another file here has been created, basic linux.this. So what if I want to see what content is present in this particular file, I will just do cat. And you can see the entire thing is copied here. See, this is too big of a file, no? Similarly, if the files are much bigger, it will go much down. This is present in that format. These files, this file is saved in .ipy something. So ipy.nb, this is the Jupyter Notebook extension. So when you open it like this, it will be shown like this. But when you open this file in Jupyter Notebook, you can get a proper file where you will have the title or if you have written. So when I open this file in my Jupyter Notebook, it will be shown the way it is shown now. Okay, the way it is shown in the background, you can see that same way it is going to be shown, so the people can understand and it will look very neat and very representable. So that is why we generally create a Jupyter Notebook file. So this is how it looks when you look at this, the code file. If you open the same file in Jupyter Notebook, you will be able to see it very nicely and in a representable way, the way you created it and the way you were seeing it before it was in a Jupyter Notebook. So that is how it will be created. So this was about Jupyter Notebook. So we'll just say don't restart. We'll just close it. So this is how it looked. This is a dead kernel. You can't do any work now here. So this is the file that it looks like. How? So now we'll just close it up from here because we have already covered it and we have already moved to the like we have already saved it in our terminal. So that is how it basically works. So this is all about Jupyter Notebook, and that is why that is how we create a Jupyter Notebook, how we visualize it, and how we differentiate between the markdown and the normal cells. Okay. So this was all about today's session where we covered the Linux commands and we covered Linux commands, then we covered Conda, how to install Conda, and then we moved on to Jupyter Notebook, how to run the bash commands in Jupyter, and then how to visualize. We also understood the difference between the markdown and the code cells in Jupyter Notebook. So that was it for today's session. So thank you everyone for attending today's session. So I hope it was clear. Now maybe we can take up some doubts if anybody has any doubts for today from today's session. Any doubts, anyone? Like I just want to ask then if nobody has any doubts. So was today's session a little overwhelming as well? Like, was it too much of technical aspects of Linux, Conda, and Jupyter Notebook? Were you guys able to understand? Maybe anybody can unmute themselves and like talk about it. So like I also understand that everybody understood the concept for the day.
Yeah, it gave the basic understanding of the softwares together, and we have to practice so it can be like for new users like me, it will be helpful. We
Yeah,
have to
yeah,
practice from the scratch.
yeah, absolutely. So I just want to ask, was the session very overwhelming? Like there was something that you weren't able to understand, or everything went good and smooth?
Yeah, it was easy to follow actually.
Okay, 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 absolutely. Okay. Thank you, Suganya. Anybody else? Anil, I saw you unmuted yourself. Do you have to say something?
Yeah, ma'am. I have some doubt earlier, but now after this session, it's clear.
Oh, okay. So your doubt is clear now.
Yes.
Okay, okay. Thank you, Tanil. So anybody else would like to share? Like anybody else, like will say the pace was the session, like was it okay? Everything clear for everyone? Can I get a quick thumbs up from everyone if everybody understood? So we can end today's session then. I know it was a pretty long session for the day because we covered a lot of practical aspects today. So maybe I'll request everyone to practice the commands and the conda things as well. So once you guys are thorough and clear with it, so tomorrow we are going to do the tomorrow or maybe in the next session we are going to cover the KIME analysis. Okay, so I will be giving the commands for installation of KIME. So I hope people who haven't installed Linux, Conda and all, so they should install Linux and Conda and keep it ready before the next session so that you can install KIME as well. And then we'll understand about KIME, what are the tools, plugins and everything related to KIME in the next session. So that's it for today's session. Maybe Vavani, we can end the session now.
Ma'am, one more
Yes,
question.
Tanil. Yeah.
Yeah, I use in mobile exam, so is it any problem?
Sorry, Tanil, I didn't get your question. You said
Yeah, I use the terminal in mobile external.
in your mobile.
No, mobile external application.
Okay. I am not very familiar with that, what you're mentioning right now. So, like, is it your
Okay.
system or your server you're mentioning?
It's a software that I communicate with the server.
Oh, okay, okay. Yeah, absolutely. That's fine. That's not like it might be a software in a server if you're using. That's absolutely fine. So, in that, we can install and we can work ahead.
Okay.
Okay, great. Anybody else? Any other questions? Or then we'll end the session for the day.
Dear participants, can you get a quick thumbs up if today's session is clear? Thank you, Tanil. Thank you. Thank you all. Fine. I think it's time to wind up for the day then. Thank you, Anshu, for having this great session on this Sunday evening. And thank you, dear participants, for joining us on the weekend. And yeah, let's wind up for the day. We have shared the feedback link in the chat. Please fill it out. If you have any concerns regarding the session phase, or if you want to have any other idea, please let us know through the feedback form. And tomorrow we don't have any session. Our next session is on Tuesday at 6 p.m. Happy to meet you all in Tuesday session. Until then, please review the session recording and study materials shared in the drive. Thank you once again, everyone. Thank you, Anshu.
Thank you, Bhavani. Thank you, everyone, for attending the session as well. I hope like we are building the steps to metagenomics together. So thank you all for attending a session on Sunday also. So all the best, everyone. We'll